In this video, we're going to be talking about the complement system. The complement system is made up of proteins that are synthesized by the liver. Let's write this down. The complement system is composed of proteins synthesized by the liver. When activated, these proteins are going to form complexes that is going to complement acute inflammation. Let's also write this down. So when activated, these proteins will form complexes that will complement acute inflammation. There are three ways to activate the complement system. It can be done through the classical pathway, the alternative pathway, and the lectin pathway. These pathways are going to start off differently, but then they're all going to converge at the same point. That is through the production of C3 convertase. The general flow of the complement system is going to be from the start of one of these pathways, the classical, alternative, and the lectin pathway, and then the formation of C3 convertase, and then we get the conversion or the formation of C5 convertase, and then finally we have the formation of membrane attack complex, which is going to be crucial for the lysis of encapsulated bacteria. Let's go into a little bit more detail on how these pathways start. The classical pathway is going to activate the complement pathway through antigen-antibody interaction. So here is an antigen. Let's say that this is a bacteria. And here we have the antibodies that are interacting with the bacteria through the Fab region. These antibodies are either going to be IgM or IgG. Once this antibody-antigen interaction occurs, in the Fc region of the antibodies, we are going to get the formation of the C1 complex. The C1 complex is important in that it can lead to the formation of C3 convertase by cleaving C2 and C4. In the alternative pathway, or the spontaneous pathway, I like to call it the C3b pathway, the start is going to be different. So normally we have organic autoactivation of C3b in our bodies that is going to be in the intravascular space. And normally C3b can be deactivated by normal cells quickly. But in the presence of pathogens, C3b is going to proliferate, and then it can interact with something called the factor B as well as factor D to produce C3 convertase. That is the alternative pathway. In the lectin pathway, we have something called a pattern recognition receptor. This is a protein that is going to bind to the carbohydrates produced by pathogens. So the protein pattern recognition receptors binds to carbohydrates produced by pathogens. Once this binding occurs, we have the formation of proteases, and these proteases are then going to cleave C4 and C2 to produce C3 convertase. The difference between the lectin pathway and the classical pathway is that the lectin pathway is not going to need the C1 complex for the production of C3 convertase. Now, after C3 convertase forms, it is going to cleave C3 to form C3A and C3B. C3A is then going to upregulate mast cell degranulation. C3B is going to do two things. It is going to upregulate opsonization, which is essentially is going to increase the phagocytosis ability of phagocytes. C3B is also going to contribute to the formation of C5 convertase. C5 convertase is very similar to C3 convertase in that it is going to produce C5A as well as C5B. C5A is going to upregulate mast cell degranulation just like C3A. Another thing that C5A does is neutrophil chemotaxis. So it is also going to upregulate neutrophil chemotaxis. Now, once we have the formation of C5B, what C5B is going to do is it is going to combine with the complement proteins C6 through C9 to produce the membrane attack complex. Membrane attack complex will form on the surface of pathogens and cause pathogen lysis. So it is very important for the lysis of encapsulated bacteria, just like we mentioned earlier. Membrane attack complex can also form on the surface of red blood cells, and it can cause red blood cell lysis. And this can lead to something called paroxysmal nocturnal hemoglobinuria. But luckily we have proteins called membrane inhibitor of reactive lysis, or MERL, as well as decay accelerating factor, or DAF, that can inhibit this process so that there's no membrane attack complex forming on the red blood cell surfaces. Another important thing to know is that MERL is sometimes called the CD59 protein, and DAF is sometimes called the CD55 protein. And that is a quick overview of the complement system. In the next video, I'm going to be discussing pathologies associated with the complement system. And this tends to be more high yield on USMLE Step 1.